time. So thank you very much for showing up for this special Cancer Center seminar series. I recognize that the series ended a couple of weeks ago, but we have a special guest today. So Professor Luca Magnani just happened to be traveling to Toronto, and we were able to get him to travel from Toronto to Minneapolis to give us a seminar. So we're really fortunate to have an international speaker of his caliber. So he's a professor at the Institute for Cancer Research Institute of Cancer Research. But prior to that, he obtained his bachelor's and master's of science at the University of Bologna. And then he moved to Purdue University to complete his PhD, where he studied developmental biology and embryology. Did a very brief postdoc stint at Michigan State. So he loved the Big Ten, and then moved to Dartmouth University or Dartmouth College to work with Matthew Lupien, who's now at Ontario Cancer Institute, and Luca moved with him when Matthew was an assistant professor. And so this is where Luca got into breast cancer research and really made a name for himself in studying the convergence of the genomics and epigenetics and plasticity of breast cancer as it pertains to endocrine therapy resistance. And so you're in for a treat. Luca's running this from his own computer because he has a lot of retro fonts and GIFs and everything like that he's worried would not run correctly on another computer. And so with that said, the floor is yours. Thank you for visiting us.
Thank you very much, Scott. Yes, thanks for all of you showing up these ungodly hours. Generally in Europe, seminars at no seminars before lunch. But I'm glad you're around. I hope that, for me it's after lunch now, so it should be okay. And I apologize already for the students. Oh, he's coming. Is all okay? No? It's the man in black.
Okay, we're the PowerPoint. Looking for the get rid of this.
Get rid of?
Let's see. Because I want to.
Can you see what happens when you do seminars in the morning?
Go back to Zoom.
So should I?
Here we go.
Never. The computer is waking up.
That's what I want to do.
Here we go.
All right, and then let's go back to PowerPoint. It looked like I quit PowerPoint. We're just having fun today. Here we go. Yeah.
Here we go. Just here. And then just there. Eyes on the other. Back in business.
All right.
Yes.
All right. And I make the pointer. Laser pointer. All right. I was just going to say that I apologize to the students that grew up in the wrong decade. This is going to be a heavily 80s derived talk. So, you stick with your Taylor Swift. I'll try to give you a bit of background on what happened 40 years ago. And the title is really like a link to the past, because the whole idea here, I'm going to try to get back at the very end of the talk, is that what is happening in cancer under treatment might be just a rebooting of some sort of developmental programs that also happen during physiological lifespan in women. And let's try to, if that will make sense. But let's start from the clinical problem. And the clinical problem for the type of tumor I studied, it is the hormone-dependent cancer. These are 70% of all breast cancer. Right? You are aware that breast cancer is incredibly common. The problem for this patient is not so much surviving 5-10 years. The problem with this patient is surviving the entire lifespan. Because as this subtype has the nasty habit of having very long latency relapse. And this is just shown in this curve over here, in which you see the top one are node positive, the yellow one are node negative. But you see that even in node 0 situation, after 20 years, 20% of women comes back. So this is very different from triple negative, in which, just to be brutal about it, at 5 years, you're either alive or dead. Here, it's almost like you're never out of the wood, and we would really love to make a dent into this problem as a lab and try to find out if we can actually get the curve to flatten out after a little while. You know, and this is people that get 5 years of endocrine therapy after surgery. And again, that is kind of an intriguing paradigm in the clinic, because generally women don't really get, or like human in general for cancer, don't get 5 years of treatment in the absence of disease, because disease is after surgery. And therapy worked very well, because if you look at the curves before surgery, so this was with radical mastectomy, a trial done, not a trial, this was consecutive patient in the 70s. And you see that women with node positive had the dismal survival. So adjuvant therapy really did an amazing job into bringing that long-term survival in, but now we need to do the last bit. We need to do the last mile, and we need to really to make sure that we eradicate these tumors. Why do I say that? Because I think this is the problem we're facing in the clinic, is that the tumor might have microdisseminated earlier than we think of. At least this is what the mouse model and a lot of like correlative measurement tells us. And what we're doing really is like, we are not leveraging enough these 5 years of adjuvant therapy. That means that, you know, there are going to be cells somewhere in the body of these women that have been bombed consecutively for 5 years with this amazing therapy. But all this therapy cannot really eradicate the disease in most of the case. But I think the window there where the cells are adapting can be leveraged for, you know, bringing really a total eradication. Why do I say that, you know, these endocrine therapy are not as effective as they should be? This just comes from the backbone of us doing as human like 120 years of the same thing. Because, you know, in the late 1800s, Bitson realized that if you take away the ovaries from hormone-dependent women, they didn't know about subtypes at the time, of course, they saw there was some sort of shrinkage in the main tumor. So they understood that the ovaries were talking with the tumor. And of course, at the time, this was not known, but this is all hormonal communication. And then in the 70s, we realized that we can kind of do the same thing with drugs like tamoxifen. They can block the activity of the estrogen receptor. So this is where the axis comes in. You have hormone activating the estrogen receptor, and the estrogen receptor promoting cell cycle in this cancer cell. And then in the 90s, we came with aromatase inhibitors. So we're doing this even better. And then we came with fulvestrant that degrades the estrogen receptor. And then we came with CDK4/6 inhibitor that are hitting downstream exactly in the same pathway. And now we have things I cannot even pronounce, you know, serum, elacestrant, giredestrant. I think they're like really having fun with us with all these names. But the take-home message here is that we're always blocking the cell cycle. We are not actively killing cells. And this is important. We'll see how this is the back to the future bit, in which, why do a cell in which you're blocking the cell cycle should die? And this is an important question. So all in all, I think in the last 100 years, we've done repeat and reboot of the same franchise, and, you know, with minimal return now. And the poor Harrison Ford, I hope he would retire, but it's, you know, it keeps going. And I think we are doing exactly the same. So why do we need to kill these cells? If you've seen enough zombie movies, you know that generally it's not that the problem. These are just there for show. They are like, you know, running, doing crazy things. But the one that kill people is the slow one. Is always the one you completely ignore, you see there lurking around, and eventually just beat someone in the very last frame of the movie. And I think this is exactly what we're doing with cell. And if we put this in a more scientific definition, I think we are leaving cells around the body. And if you leave a cancer cell around, it will evolve. Doesn't really matter what you do. So you need to kind of get to surgery, but we cannot do surgery on things we cannot see. So this is why the tricky bit with systemic therapy is. There's a lot of unexplored possibilities. So this is kind of like where people, I think, has improved now. But like 3 or 4 years ago, this was the paradigm, right? There were heterogeneity within the primary tumor. There were some cells that have some sort of mutation that will make it resistant to second-line therapy, first-line therapy. The only one that we really know something clearly is the receptor mutation. So when the estrogen receptor gets mutated, now it doesn't need the ligand anymore. And of course, the tumor is estrogen independent. But I think this is kind of an oversimplification. And we know, for example, that there's only 1 to 2% of women that have estrogen receptor mutation at the onset of disease. So a lot of things are happening in between. And to be honest, we don't really know what's happening in between. We just know that a lot of this effort has been done on sequencing effort has been done on the primary side and the metastatic side. But we know next to nothing of what happens in between. And again, that between could be 5, 10, 15, 20 years in which cells are microdisseminated in the body. They're under the pressure of therapy, but we don't know how they evolve and what's happening in there. So there's a big fat question mark over here. And the lab is really trying to learn something about this. And of course, there are limitations because this microdisseminated disease is invisible. So we need to model it in other way. And one of the first things we've done is looking at a rare cohort of women that don't receive surgery. So the general practice here is that if you are a hormone-dependent patient, you immediately go to surgery. And generally, this happens, at least in the UK, within 28 to 30 days. And then you get these 5 years of therapy in the absence of any MRI-positive tumor. But in this case, these were 5 women that refused surgery. So they had the local tumor, and what they accepted is just to be treated for long-term with this endocrine therapy. So this is a great model because it will tell us exactly what the therapy is doing when you give it for years and years and years. And you can see the response here. So some of these women had response for 4 years. What happens generally, you can shrink the tumor a little bit. Differently from triple negative, you never get to a complete response. So you never eradicate the tumor, but you can shrink it quite effectively. And then what happens is that some of these women had a progression. So this is not a relapse in the sense, it's a progression because the tumor mass is always there. And this is just what happened under therapy. And what we've done is whole genome sequencing pre and post progression. And this was 120x. So we spent, at the time the lab was just starting, I think I spent 50% of the budget on this experiment, and we found nothing. And that means that we couldn't find a single mutation, either coding or not coding, that really explained why the tumor became progressive. Everything was pre-existent. All the classical mutation, you see them here. The driver were in there, but nothing acquired really explained why the tumor now went from not proliferating to proliferating. And so, this kind of led us to think about exactly what's happening here. In this phase, in this particular time, dormancy was modeled in these 4 years in which the tumor shrank and was maintained. So what is happening there? What is really driving this progression from a non-proliferating tumor to a proliferating tumor? So we kind of call this dormancy. A lot of people will think this could be equivalent to senescence. It could be cancer stem cell. There's a lot of semantic in there. But what I think is important to think is that if cells don't proliferate, the likelihood of acquiring new mutation is dwindling. So you need to proliferate if you want to fix DNA damage to a mutation. So what is really getting a cell that is not proliferating out of that state? And if you are a developmental biologist, you don't need a lot of convincing, but if you are an oncologist, I think you need a bit more convincing that you can change phenotype in the absence of mutation. And this is, of course, what epigenetic is. So what we really think is that there is epigenetic changes in these non-cycling cells that happen over time. So how do we study this stuff? And of course, here I take the Jack Nicholson, if you watch The Shining, it starts pretty normal and then it just gets deranged. And if you don't know, like if we do like things we do in science, and we take the snapshot at the start, snapshot at the end, and you need to make a story, you will never really learn what's happening here. So we try to think about a way in the lab to model this at much higher granularity. And the principle was like, first of all, we want to avoid perturbation. So as in these women that had a primary disease and they didn't receive surgery, but only endocrine therapy, we wanted to model exactly the same. We wanted the tumor mass to stay there while we were putting constant pressure. And this means we didn't want to split cells at all. So we started with very large flask. We wanted to rerun the experiment multiple times because, this is something that you cannot do with human. We don't really know if you were to clone the same woman and go back in time and rerun evolution, would that always go in the same way? And there's a lot of indication from kidney cancer, for example, that the tumor does not evolve every time the same way. These are experiments done with people that have a predisposition to getting cancer, and when they realize that the same person gets cancer all the time, consistently, and every time in the same locus, if we look at what's happening, the tumor evolves every time differently. So there's a lot of stochasticity built in the system. And then we want to study non-genetic modality of evolution. So we really want to study the epigenetic. And the key thing for us was just to be patient, because evolution doesn't work in 10 days, 15 days. Evolution takes a long time. So here we're trying to model something that takes 20 years. And if we really want to kid ourselves, we are going to do a mouse model that in 10 days will give us a result, I think we're not up for anything meaningful. So we assembled the team. This is the team. It's Dalia, Emre, and Hina. Hina is the computational biologist. The other two are the wet scientists. And when we started, we had this great expectation, right? Miami Vice vibe. We just needed the Ferrari parked outside. So that was the spirit. And we didn't know that this would have taken us 5 years And it became this, it became an experimental swamp. If any of you have ever watched this movie, you have terrible memories about this, but this is the swamp of sadness. And this is what happened to the lab for a couple of years. That is me, and the horse are the poor postdocs. So I had to drag them out. In the movie, they die. In this case, they didn't. So I managed to get them out. And this was the experiment, the main experiment. So we call it tradition. And what we've done is really leverage technology that was new at the time. These are transcriptional barcodes. So it means it's your classical genetic starting barcodes. That means you can give a name to each cell. But these are also expressed as a transcript. So that allows us to measure things in single cell experiment as well. And you'll see why this is important later on. So we basically created a pot, this massive progenitor flask of 90 million cells, in which each one had just one barcode. And then we randomized that as it was a trial, because remember, we want to rerun this multiple times. And we created 56 carbon copy. We certified, we did a lot of work to show that each carbon copy was the equivalent clone of the other. So the barcode representation was exactly the same. The frequency were exactly the same. And then some of these flasks have been treated, put under pressure, either with tamoxifen or aromatase inhibitor. So here, simply, we take away estrogen from the flask. These are 20 liters flask. So it means that in each incubator, you can put a couple. So we took over the entire department. And this was good because COVID was around. So the department was abandoned and we could do all our crazy stuff. And then basically we started to follow this hyperflask in time. So we were waiting, then they went into dormancy, so they shrink, exactly like would happen in women. And then you shrink them to a certain point, and then we waited and waited and waited. And each time we measured everything. So we measured the barcode frequency, the genetics, and nanopore. I'm not going to show that data at the moment. RNA sequencing, histone, mass spec, the kitchen sink, because of course we didn't know what to expect. And I think this is always the first and the most intriguing finding we had at the time. Imagine this is just measuring how long was this movie. And we thought these are carbon copies. So the system is deterministic. This is the most simplification we can of a tumor because this is a cell line effectively that is coming from just one clone. So how difficult could be to model this? And basically what we saw is exactly what you see in women, that it takes a while for the cell to respond to therapy, and then eventually the tumor shrinks. And when you see in yellow, it's just these cells that are in dormancy. So this is the number of cells. And then when we waited for this progression, but the progression was completely stochastic. So we had some flasks that woke up after 90 days and some flasks that were just waking up after 6 months. And just, for the curiosity, we had one flask that we had to terminate because we moved institution and was still dormant after 14 months. So it means that it's very hard to predict, even in completely what you would imagine is a replicate, what would happen to your system. On the top, you have MCA7, and on the bottom, you have T47D. So these are two different models. The bottom one is a P53 mutant, but the story is exactly the same. As I said, this was surprising because we thought these were all clones. Like once you figure out what happens with one, everything will happen exactly the same in all the other flasks. But of course, this is not the case. These are just alternative flasks, right? This is not a demonstration that this is really true if you were to run the same thing and you were watching it. So what we've done is a similar experiment, which we took over incusites, and we started to look at this flask for month and month, taking 6 pictures every day. So people, again, hated us because we hogged all the resources for a very stupid experiment, if you think about it. But this is what happened when you look at the individual awakening events. These are exactly in the same spot. And you can see that you have some wells that woke up very early and some wells that woke up very late. And these, again, show you that a couple of wells were actually still dormant at the time of finishing the experiment. And one well was eradicated. So for some reason, one well, we could cure the cancer. Very intriguingly, when you have this image, we realized there were things that we label field awakening. And I'm going to go back to this later on in the talk. But it means that the full progression doesn't happen once. This is almost like you're starting your car. So these flasks were trying to do something. And you can see, for example, this colony over here. that at day 97 became quite big. And we thought, okay, that is a waking up event. It's fantastic. But then you realize that the cell eventually regressed and went back into dormancy. So the full progression is something that happens rarely in the flask, but it doesn't happen only once. And this is complicated, you see, because for experimental point of view, if we want to map exactly what's leading to the progression, we don't know if we are capturing a real progression or a field awakening. And we're doing a lot of biology to try to understand this now at the moment. But the other bit that was important is that we realized that the full awakening, so the one that really leads to the final expansion, only happens from one spot in the flask. So this is important because associated with the barcode analysis will show that basically the full progression, the clonal sweep, is just from one cell. So what happened, like in terms of heterogeneity? We look at these are the various flasks, and we call them with Greek letters. And the intriguing thing is that they don't respond the same way to second-line therapy. And it's very hard to predict what a flask will do in one case compared to the other. And I'll give you just this example. If we go and treat them with fulvestrant, that is the standard of care for these women. So after they relapse, after CDK4/6 inhibitor, they get a degrader. And you can see that 3 flasks here responded very well to fulvestrant, but 2 flasks were de novo resistant. And if you look for the palbociclib, it's exactly the same. So it's very hard to predict when the flask wakes up, what will be the phenotype. And I'm not showing here for the interest of time, but the morphology of this awakening was completely different. So for us, this was important because it's really telling us that, if you go in the literature, you look at MCS7 modality of drug resistance, there are about 300 different mechanisms. And everybody claims that there is the right one. And I think the problem is that they're all real. And it's just really depending on what happened in that particular experiment. So the way we think of this is, again, you get 3 different endings every time you run these movies. It's not something we can really predict. And if this holds up in the clinic, it's problematic because the standard of care is just one for everybody. It might work in certain cases. It might even work with certain lesions, but not all of them. So what about the barcodes? So we chuck in 100,000 barcode, and we have done the experiment quite well because in the pot we found exactly 100,000 barcodes. So the postdoc were amazing in the skills. And then when we start to apply all this therapy, you can see that the number of barcodes representation is reduced with time. And when they wake up over here, you realize you're in the order of 10,000 barcodes for tamoxifen and 5,000 barcodes for the aromatase inhibitor. So it means that when there is a full progression, there's a lot of lineages still in there. That doesn't tell you much, and I'll show you what the frequency means. But what we've done, what this is important, is that the fact that there are still about 40% of the lineage there, beg the question of, is there something predisposed that will tell if a cell can enter dormancy or die? Because of course, 60% of the lineages are eradicated by the treatment. And we've basically done this experiment 100 times, in which we reduced the barcode complexity, but we ran the experiment 100 times. And doing that, you can start to do mathematical modeling. And just for keeping a long story short, what we discovered is that the entrance in dormancy is completely stochastic. So there's nothing at the lineage level that can really predict, or nothing we could measure that could really tell us if the cell was biased towards entering dormancy or dying. It's just a number game. And again, that's problematic because, what we really want to do is want to make sure that this therapy kill all the cells, not just 40% of them. So then we look at these awakening lineages, and this is where the surprise came in. As I said, there are still thousands and thousands of barcodes, but if you look at the frequency, you realize that 99% of the cell in your progression is basically one barcode. So there was a massive clonal sweep in all these experiments in which you have now 50 million cells, and 50 million cells, 99% carry the same barcode. And if you put this together with the imaging, this is kind of showing that the final awakening just came up from one cell figuring out exactly what to do and carry over. The tricky situation is that the barcodes are all different in all the experiments. So it means it's not the same barcode at the start. This argues that this is not a pre-existent phenotype. This is something that is probably acquired and happens stochastically at the awakening level and drives the entire phenotype. Again, so this is pointing out this is probably not pre-adapted, is not pre-existent, and I'll show you it's not probably genetic. So the situation you need to have in mind is that now you're in this bit here. The clones, only one clone awakens, But what I haven't shown you, and I'll show you later, is that all the other clones in the flask are dormant. And if you wait long enough, another clone will wake up. And what we realized that the other clone will have a completely different phenotype from the first one. Again, we could learn this simply because the space in this flask was enormous, but it's something that otherwise you would not be able to capture if you were just doing experiment in the classical way. I told you that this was not genetic, and I'll give you the data. These are like phylogenetic tree based on whole genome, and we gave this blindly to our collaborator that is just a genetic guy at the ICR, Andrea Sottoriva. So he looked at this and he said, you know, look, these are tumor evolving independently from each other. And when I told him that this was exactly the same line, he was a bit shocked, but it fits with all the barcode analysis, of course. And this is telling that the generation of resistance came independently in each flask. Now, these are the whole genome mutation. And remember that, so what we've done here is compared whole genome at the progression with whole genome at the start. And if something expanded, remember, this is just one barcode, it should have been pretty obvious. If there was a mutation that was responsible for driving that phenotype, now goes from very rare to 99% of the population. So there's no way for us to have to miss it. And the only thing that was vaguely intriguing was this ER alpha mutation, the H524L, that came just in one flask of the tamoxifen treated. And this is quite a rare mutation that people have observed in tamoxifen treated patients, so it makes sense, but it's only one. And the interesting thing is that this is what happened when the tumor progressed. So this is the allele frequency. It was quite high, about 50%. But that allele frequency is counter-selected if you then let the cell proliferate. So it's also arguing that this was the actual cause of the progression, because you would imagine that the barcode would consolidate in time, but the actual mutation is counter-selected. So it's just simply saying that this is most likely was not the cause of resistance. In all the other cases, we couldn't find anything. We have done the same experiment with T47D. Again, you can see that this is just one barcode taking over, and there's no mutation in there. So there is one clone taking over the entire population, and there's nothing at the coding level. We'll look at the non-coding level. We'll look at everything. Nothing genetic that could really explain this. Then, of course, we thought, you know, this is a matter of granularity, so let's go single cell. Remember that these barcodes are transcribed. So it means that if we do a 10x single cell genomics, you can capture the RNA-seq profile of these cells, and you can capture the barcode information as well. And in our very naive head, that means that we could look at lineages, right? So we can look at the Luca cell at the start, Luca cell at the end with single cell RNA sequencing, and really capture the transcription or reprogramming. And I'll explain later why this was a bit of a naivety thought. But this is the experiment. So we reduced the number, the barcode number to 100, because we know it's stochastic. It doesn't really matter if we have 100,000 or 100 or 2 for that matter. It's just a question, how many times can you replicate this? So we put 100 barcodes, and the reason for it is that 10x is limited by the fact we could capture 10,000 cells. So we didn't want to have risk of sampling different barcodes and not being able to put them linearly. And we've done the same experiment, as you can see here with the IncuCyte, that after 5-6 months, there's just one region of the flask that wakes up. So if you see purple, it means that that particular colony took over and then eventually took over the entire flask. The barcode is again just one. So out of these 100 barcodes, just one takes over the entire population. And then we have, of course, done single cell RNA-seq, and what we discovered the US. And this is just the phenotypic map, as you can see here, that is quite plastic. But now the question is that, how can we dig into this nice data set? These are about 500,000 single cell. So the question was like, are these completely plastic? Is this phenotype associated randomly with all the treatment, or is there some sort of logic into this? And what we realize is that, you know, cells start from New England. You can see over here in green. They are very well educated. You know, they just came over from England. Their accent is perfect. We know what to do with them. And eventually they start to be hit by this winter. And of course, they go to Alaska. And the phenotype is quite reproducible. And I think this was a plus. It just means that out of these, you know, stochastic entrance in dormancy, the lineage is stochastic, but the phenotype is not. So all these cells from different lineages, different genetic background, whatever you want to think of them, they are quite heterogeneous. They all go in the same spot. And I think this is intriguing because it means that we could target eventually that spot. This is where the optimism comes from. There is a place where these cells go that we have not studied in real women, but they might carry vulnerability that we could be able to exploit further on. Because this is what happened when they wake up. When they wake up, they recolonize the entire continent. And remember that it's just one barcode. So one barcode could evolve plastically in all the other phenotypes. And this is annoying because I showed you already that all this plasticity means we cannot really predict what they will respond in second line. We cannot even predict their morphology, and we cannot predict when this will happen. So I think our goal is now to hit them here. We should not wait these states because it's good luck to try to target all these different phenotypes. But what drives this? And I think this was the million-dollar question. And we knew it was not DNA methylation. And again, I cannot show you the data for an interest of time. So we thought, of course, this might be epigenetic. It might be some sort of histone modification regulating transcription. But without knowing what that is, we went unbiased. And we did a super-SILAC mass spectrometry. Sounds very cool. Is nothing else than, a very precise way of measuring histone modification in combination on histone. And what you can see here is a list of things that might tell you or not tell you anything. These are all these modifications that are associated with this histone code. So you have, for example, H3K4 trimethylation that is associated with active promoter and all the rest. And what we realized is that there was a series of modifications that were quite highly enriched in dormancy. And this was reproducible also in the second model. And just to kind of summarize this in one slide, what we realized is that all the dormant cell ended up accruing a lot of heterochromatin. So a lot of other modifications were quite all over the spectrum, but these three histone modifications that are associated with repressive heterochromatin were quite consistently reproducible. So we thought there might be some biology in there. And so, how can we test this? So the first thing we look is at the RNA-seq spectrum. And again, I said that you already saw this at the single cell level. I mentioned that there is just one dormancy phenotype. There are not multiple. And this is kind of quantified over here. So this is a PCA plot of this RNA-seq. And what this is telling you is that when you treat cells for a long time, they all go yellow, right? So this is the dormancy bottleneck. So the RNA profile is very, very, very conserved. But when they wake up again, you remember all this heterogeneity. This again is, we can see it even at RNA-seq level. And for all the people, this is for the aficionados of cell culture. You go in the cell culture and they tell you don't culture yourself for more than 20 passages because otherwise they go mad. They don't go mad. So this is what happened. The untreated were here. These were cells treated for 6 months. And we kept splitting them because this was the non-treatment side of the trial, and you realize that they don't move much. So cells don't really evolve in plastic. They already figured out what to do, and they will keep doing that if you don't really annoy them. Single cell level, as I said, we can quantify this differently. If you see these are all the cluster we call in the North American landscape. But what you realize is that in dormancy, they have access to a couple of them. These are the yellow Alaskan signatures. So it means that they don't do dormancy in many, many different ways. They do dormancy in just one way. And remember, this is all in the spectrum of this heterochromatin context, because it's going to become very important to relate the heterochromatin to this RNA profile. Is this, I only show you cell line. I kind of want to put this right here. This is preliminary, but we are doing this at a much better level now. This also happened in women. Do you remember these 4 or 5 women that refused surgery, and we couldn't find any mutation in there? So what we've done with this sample was to use spatial transcriptomics to only map the transcript of ER alpha positive cell. Because the problem when you do old school RNA-seq on this sample, you that smash everything, adipocyte, epithelial cells, and all the rest. And of course, you're going to end up with a signal that is epithelial because you have killed, the only thing you're left is with a tumor. But at the start, you generally have a very heterogeneous tissue. So to solve that, we took 12 ROI, so 12 area of interest. So this is a multi-region RNA-seq. At the start and at the end. And you can see that at the start, the phenotype is quite consistent. So they are almost overimposed one on the top. But when the tumor progresses, you start to have all this spreading of phenotypes. So what we saw in the cell line with that RNA-seq that was clearly showing you that the cells were very consistent at the start and very spread at the end, also happened in women. And the intriguing thing is that if you look at the tumor of these women, and this is probably not very evident from these small slides, is that in the progression lesion, there were pockets or area that were still dormant. And this is very reminiscent of what we saw in the cell line as well. A lot of lineages were dormant. Only one lineage took over. And we think that this is exactly happening of course, at scale in these women. Now, this experiment was just done in four women. So we're repeating this with a cohort of 60 women in which we have also intermediate data points. So we'll see if this holds true. I mentioned, remember the transcription heterogeneity. That is reflected partially in the epigenome. So this is H3K4 trimethylation. So this is an histone mark that you put on the promoter of active gene. And this is pointing out that this plasticity is also partly heritable, because once you start to kind of put all these flags at the promoter, you know that the gene will stay on and will be transmitted as being on to the other cell line. But this was promising also because it showed that the dormancy, again, was quite reproducible. So dormancy seems to be a true reprogramming event. It's not just a response to therapy. It's not a transcriptional response. It's really shifting the epigenome from one set of gene expressed to another. But look at what happened with the K27 trimethylation. So this is a mark of repressive. This was one of our key marks. And these are the day zero. These are the awakening. And these are the dormancy. So not only, and look at the PCR variance. So 94% of the variance is down here. So don't be fooled into thinking these are different. These are basically exactly the same thing. I forgot to tell you, these are cat and tag experiment, of course. This is important. And this is meaning that we could take the representation. We know with mass spec that the heterochromatin was conserved, but we didn't know where that was in the genome. And what this is telling you is that the K27 trimethylation is all at the same loci. It's not widespread across different regions of the genome. For the second time, again, I cannot go too much into detail, but this is showing you that this massive heterochromatin is repressing consistently the same transcriptional programs. So cells are going into a corner, they're a bit scared, they're a bit stressed, but they are all stressed in the same way. And this is also an inheritable cell state. This is not simply plasticity or transcriptional reprogramming. This is truly a different cell state. So just to summarize what I told you so far, we think there is a massive reduction in the epigenetic state or epigenetic heterogeneity in these cells. And we think that this is the time which we should really hit them, because they are not, they are stuck, they're not plastic yet, they have just one phenotype, but we know next to nothing about what is really the vulnerability of this state. One critical experiment that we've done here is to show that this is functional. This is not just a fiction of a figmentation of my head. So what we try, you can imagine if there is a truly epigenetic reprogramming, you expect that these modifications are important for the dormancy state. To test that, what we've done, we took every possible epigenetic inhibitor in the market. Some of these are like tulminostat. This is a H3K27, is an EZH2 inhibitor. And this is in clinical trials for lymphoma. So that means that this is great because we could eventually go quicker into phase 1 trial in breast cancer. And you can see that these inhibitors that are targeting, again, this epigenetic writer, they don't do anything when cells are untreated. So when cells are happily proliferating with estradiol, you can give them micromolar of this stuff and really nothing happens. The second experiment what we've done is just, of course, we send them into dormancy. Remember, to do this, we need to have long-term endocrine pressure. And in our cell line, that takes a month. This is not something you do in 2 days. In women, probably we don't know how long it is, but from neoadjuvant trial, we think that this could be 6 months to 1 year. So we were a bit discouraged at the start because, again, we take off estradiol, we add the inhibitor, and we thought we're going to kill everything now. It's solved, Nature paper. And 7 days, nothing. Absolutely nothing. But look what happened after 28 days. So in a lot of cases, we could eradicate almost completely the dormant pool. So it means that if you keep the pressure up, you need the epigenetic reprogramming, but this is not something instantaneous. And remember that a lot of screen companies do, a lot of these things are generally assets that you do in 2 or 3 days. And you want to see a single agent effect at 2 or 3 days. This is pointing us that you need 30 days of a double combination to start to see an effect. And I think this is also why probably we haven't picked up these things prior, because who is idiot enough to kind of do 30 days experiments with things that don't look like working. And this is the rationale. I think this is proving that the epigenetic reprogramming is really important. And you need to kind of block that process if you want to block cells entering into dormancy. And this is the key. I think if we can do that and we can exploit this sort of paradigm in women, and remember that, generally women don't receive neoadjuvant treatment before surgery. So we don't tend to do a lot of endocrine therapy before surgery. We do it after. And the problem is that if you do it after, then you cannot really monitor anything of this. We really need to move to a situation in which we do 6 months of endocrine therapy before surgery, and we start to try all these drugs.
Luca, did you ever leave the estrogen in?
So we've done a lot of drug holidays. And so the intriguing thing is like if you do drug holidays, especially with the dormant thing, what you're going to end up is the same phenotype. So you always have one barcode at the very end. But what happens is that if you have been dormant for a month and you give estradiol back, they take 15 days to wake up. If you do 2 months of treatment and you give estradiol back, it takes them a month to wake up. So we think there is a cumulative reprogramming there, and it's very hard for the cell. But this is why I say this is not simply a reflection of the estrogen deprivation changing the phenotype. This is a true epigenetic reprogramming. So this is preliminary again, because finding this data set is quite complicated, but these are the prognostic significance of this finding. If you look at women that don't get any, no endocrine treatment, these of course were all the placebo arm in this very old trial. And you look at the expression for these 3 epigenetic modifier. Remember, these are EZH2, EAH, G9A. It's just the name is the one that catalyzes K9 dimethylation, and the KMT5C, so H3, H4, K20 trimethylation. If you look at the expression significance in untreated women, nothing. But look at what happened if you look at women that only receive aromatase inhibitor. Take this with a pinch of salt because the number are extremely small. And these have to be extrapolated from these larger trials. But it seems to suggest that if you are very low expression for whatever reason of this epigenetic writer, and you treat women with endocrine therapy, you almost never see a relapse. This is kind of pointing to the fact that if cells don't have the enzyme expression to really enter into dormancy, they don't have option. Endocrine therapy will eradicate this. But if you have high expression, so these cells can effectively go into dormancy, this is when you start to see this very late relapse. Remember, these are 200 months. This is almost 10 years. So it's not good enough. I think we thought, okay, this is great. It means that we probably have an angle to enter into prevention of dormancy, but can we target already dormant cell? Because imagine you're a woman at your 2nd years of endocrine therapy, and we can tell you, oh, we have maybe some solution early on, but we don't know what to do with you. So we tried to try all this drug into cells that were already dormant. So it means that we did 30 days pre-treatment and then enter with the drugs. And only G9A seemed to be effective here. And the intriguing thing is that if you look at the target, this kind of introduces a paradox because dormant cells don't have expression of these genes. So once they enter into this heterochromatin state, they completely lose the driver. And part of this is because these drivers, these epigenetic writers are cell cycle regulated as well. And they're generally low in G1. And these cells are, of course, dormant in G1. So we thought, how could it be that the drug is killing these cells? I mean, it takes a long time, right? It takes about 30 days to start to see an effect. But how is it possible if the target is not there? So we thought this is all side effects. Let's Nature paper, close down, +1. And then I think we realized that there is something intriguing there. And we learned that this goes back to the field awakening. Do you remember this business of cell trying consistently to come out from dormancy, proliferate for a little bit, and then losing it? So they proliferate for a little bit, and then they kind of go back to dormancy. So we started to model this with endogenous reporter. So we have a FUCI system into the cells in which we can do basically 6 months of confocal microscopy. So another brutal thing to do, but people stick in the lab, so they must be happy. And you can show here, what we show here is that there's this bubbling, right? So cells are quite dormant for a while, for a few, like 30, 40, 50 days, in which you literally don't see cells doing anything. They are stuck in G1, and you see because there's the absence of geminin. And then you start to see this bubbling. And we really think that this is quite similar to a volcano. When you see a volcano erupting, that is your progression, but there's a lot of earthquake before that. So there is things that are trying. And I think it's also demonstrating that just re-entering the cell cycle is not enough to acquire resistance. You need to have something else. So you need to proliferate, and you need to be able to maintain that proliferation. So when we look at the single cell data, I think we got an inkling of what was happening. This is G9A, as I remember, this is the only one that can kill dormant cell in this initial experiment. And is quite highly expressed in untreated cells. And here we also separate them in G1, S, and G2M. And we could do this because, of course, single cell allows you to really capture a feeling of what's happening in this data set. So when they're in deep sleep, as we expected, there's no expression. So G9A is gone. But then they get in, restless and these nightmares. So this is where these failed awakenings start to be happening. And look at what happened in G2M. G9A expression goes up again. So in all these cells that are trying to do something, now your target is re-expressed. And we think that this is, and then of course in awakening is there. So what we really think is what's happening is that you could target dormant cell, but you need to be patient because you need to wait for the cell to go through this continuous failed awakening. And every time they go there and you remove G9A, they cannot really, they don't have alternatives. So they cannot re-enter dormancy. So slowly we are kind of eradicating them. Again, this is a principle we hope to improve on, and we need to go in vivo. We're gonna do this in mouse model now and in rats model because mouse model are crap for year alpha biology. We can say crap, right? It's not gonna get edited out. But I kind of the last 10 minutes, I want to give you some more new data, and I want to go back to this, back to the future. So I'll summarize what I told you right now. This has been published a couple of months ago in Cancer Discovery, so you can find it there. And this is what I just said. You have this constant pressure creating this dormant cell, and we really think we could block this transition early on and try to eradicate these cells. This is just, again, for the 80s. If you don't know this band, you should check it out. But we are just at the start, and I think we really need to do much more in this spectrum. And one of the things I want to tell you is that this is not just cell line work. So in neoadjuvant trial, this is neoadjuvant that we've done with a hospital in Cremona, where they routinely give 6-month therapy before surgery. This is just the only hospital I know in the world that does this. But when we applied RNA-seq, we could see that after 6 months of therapy, our dormancy signature is through the roof. So something we capture in cell line is recapitulated in women. And with single cell data, again, these are not published. These should be out anytime soon now. We could show that in single cell, in second line therapy, you can go back to this dormant phenotype. So this doesn't happen once, this happens more. And this is just shown basically when cell woke up, we gave second line therapy. And what you see here with palbociclib, they reacquire this dormant phenotype. So it's something that probably we could exploit also in second line, third line. And this is great news because it means you don't have only one shot into targeting this dormant cell, you might have multiple shots. But the key thing here is that if they get an ER alpha mutation, that this game is out. So if you have ER alpha mutation, they don't enter into the dormancy bottleneck. They don't care whatsoever. At that point, they won the lottery, and that is a different disease. And I think there we need to go with really strong SERD, but we cannot reapply these ideas. How can we stop adaptation? I just told you epigenetics, so I'll give you like a couple of vignettes of other things. Epigenetic, of course, is interrelated with everything else, and if you enter in this loop, you will never get a job, so don't do it, please. Just pick one of these 4 things and study that. What we've done here is one of the first CRISPR screen to really see how to target dormancy. And I keep this short. So I think at the time we didn't know enough and we'd done something quite naive, but the idea of this screen was to really look at the cis-regulatory elements. So we target announcer, at TAT boundaries and all these fancy things, but we didn't really think at the gene level. And what we wanted to do here is to try to find out if we could block the activation of announcer to prevent dormancy. And after this massive trial that we've done with Novartis, Giorgio Galli, and Eros that now is in Vienna, we came out with one hit. These were about 120,000 sgRNA, was again a massive screen at the time, but the only thing we could capture was USP8. And I'll show you quickly how that works. What we were very successful in finding out is how can you get cell to ignore completely dormancy. And basically this is an NF-kappa B pathway. So we realized that if you take away all the toll receptor pathway, cells completely forget about the stress imposed by endocrine therapy, and they happily skip dormancy. And again, there's more information for that aspect of the story in the paper. I think for the interest of time, I cannot go more. What I wanted to show you is that this is, how we validated USP8. And this is done in a lot of cell line right now. And again, you see, if you take away USP8 with now a genetic approach, so a CRISPR knockout, nothing happens. But if you start to do this in conjunction with endocrine deprivation, now you start to see very strong phenotype. And this is very reproducible to the point that, remember, there's a lot of stochastic entrance in dormancy, but most of the plates were eradicated by USP8 treatment. This can be found in this paper, but the reason why I wanted to mention this is because of the fact that dormancy is a problematic aspect if you want to study with CRISPR screen this sort of biology, because there's a lot of stochasticity in the process. So this was our first replicate in the CRISPR screen, and this is our second replicate. They completely diverged. And that means that if you have a guide RNA that decides to hitchhike on a cell that goes into dormancy, it will look like it's enriched. If it decides to hitchhike on a cell that will be eradicated by the treatment, it disappears. And that creates a massive baseline noise. And this is shown over here. So these are all non-targeting sgRNA. And these are massive signals. So your baseline is all over the place. And if you want to try to capture a signal out of that, good luck to you. And this is why the screen probably didn't give us enough data point, because just the baseline was absurd. So to avoid that, what we realized is doing this another screen that was a bit mental, we've done with AstraZeneca. This is now targeting epigenetic modifier transcription factors. And what we've done here, we leverage doxycycline-inducible CRISPR screens. But we had to work so hard to get this tight, because what we wanted to do is first get cell line into a dormant phenotype and then trigger the screen, because it means that now our baseline would be much more interpretable. And after we've done this, it took an immense amount of work from Hannah. We came up with some intriguing findings. I'm going to go very quick because these are confidential, so please don't repost. I know it's registered, but please don't run away, because we're going to outcompete you. And some of these is what you expect. So these are sgRNA that only targets dormant cell. They don't target dormancy in dormancy entrance. FOXA1, everybody knows about this in hormone dependent. But if you ask me, and I've been in the field for 20 years, about some of these targets, God knows what they are. So I think there's a lot of hidden biology there in which the vulnerability for this dormant cell are completely different from what you find at the start and at the end. And all we've done so far is characterize in the start and the end. So hopefully one of these targets will be real. So we are validating all of them. So now we are through with the cell line work, but we want to validate them in a mind model. But we think there is something here that might be intriguing and might be helpful for patients. And now in the last 5 minutes, if you're still awake, I want to tell you what's happening going forward. I don't think with single cell, we're going to get away from the stochastic phenotype. So what we're engineering is system in which we can image evolution. And we're working pretty hard here because you need to hit the sweet spot between being able to image 1 year of evolution and do that in enough resolution in which you can track cells. So that is not an easy thing, because in the technology we're in the lab, they are great. Either you do, HT, high throughput imaging, or you do long IncuSite. There's nothing in between. So we're building microscope to do this. But one of the funny things we found, and I think is an interesting finding, is that, remember, we have this Fuji system in which basically we can tell if the cells are in G1, G2, or S during our evolutionary adaptation. And we can track these at single lineages, right? So we can look at the single cell and reconstruct the movie of where the cell was during this period. And this is the phylogeny that you get. So you get when a cell divided, how long was in a cell cycle, how long was in G1, how long was in G2. So you have the ID card of all these cells. And the movie is played over here. And I don't know if it's going to move. And you can see basically we can track all these things. And what we realize is that, you remember I told you that the entrance in dormancy is completely stochastic? I lied. There is something that is a bit biasing and we could really learn something from, and that is the initial cell cycle stage that the cell are when they get hit with endocrine therapy. And this, I think, I was very surprised by this, and I'm very intrigued. So basically you can see with a lot of treatment, the cell cycle stage at the start doesn't make any difference. But for aromatase inhibition, you have twice as much the effect. It means that if you're in G1, when you are hit with estrogen deprivation, you're almost guaranteed to go into dormancy. But if you're in G2, it's too late to pull the brake. And we are really now spending a lot of time to try to understand the mechanism. But this is intriguing because, in a fantasy world, means that if we could synchronize the tumor of these women before we end up with endocrine therapy, we could probably eradicate many more cells than we can do right now. And this is one way of thinking about plasticity that generally people don't think about. We think about plasticity in terms of transcriptome, epigenomics, and all the rest. And we don't have to thinking about the most plastic thing that there is that the cell cycle. Cells go through it all the time. And that is why also cell lines are heterogeneous, even if you don't think they are. Last 2 minutes, going to Back to the Future. Is this relevant for tumor prevention? And I'm just going to give you 2 slides here. And what we've done working with these 2 excellent pathologists is to thinking about how this is happening in real physiology. And you might be aware that, the breast is not always on. So the breast is off for the first 12, 13 years of a woman's life, then is on, and then goes off in menopause, right? And we thought, this goes back to the idea of if a cell doesn't cycle, why should it die? If that was the default modality for the ER alpha positive cell, you would not have that lactating kids because all the ER alpha positive cell would die in pre-puberty. And so we look at just the area, the proportion of ER alpha positive cells in the breast of aging women. And what we realize is that the breast of a postmenopausal woman is basically made up only by ER alpha positive cells. All the ER alpha negative cells are gone. And this goes against the dogma. We thought that this was not going to be the case at all. Because we know that the majority of the ducts are ER alpha negative, and the dogma is that the ER alpha positive are talking to the ER alpha negative to tell them proliferate in a menstrual cycle related fashion. So we know that this will happen, right? So now you have all these cells that we think they are retired. So they don't have anything to do. The hormones are gone. They're all ER alpha positive. They're refusing to go anywhere. They're sitting there. But as I said, if you leave something to evolve, eventually will evolve. We look at men as well, and the only reason why we look at men is because I wanted to post a picture of Rambo. But basically, men's are postmenopausal women for life. So we have ducts, they are very primordial, but they're all made by ER alpha positive cells. So there's no ER alpha negative cell. This is not a functional breast. So, and this is intriguing because, this is Ki-67 over here. We never see proliferation as well. So we think there is something in there. So cells, ER alpha positive cells know how to survive in low hormone dependent fashion. And this is something they know how to do independently from oncologists or everything else. So we've done spatial transcriptomics, and what we realized is that the signature in old women completely overlap with the dormancy signature we see in cancer patients. So it basically means that these retired cells have a lot of common feature, we think, with cancer cells that are under endocrine therapy. If that's the case, I don't know. So again, we're scaling this up with a lot of donors right now. These are not easy data set to get. It took us 2 or 3 years to find the right cohort. But if this confirms, I think we have an handle in the possibility of eradicating ER alpha positive cells before they transform. Because you can imagine if something can kill a dormant cell in cancer, hopefully will translate in killing a retired cell. That's the dream. I don't know if that will ever work out, but this is exactly what we want to do. And this is important because if you think about the incidence of breast cancer, skyrockets between 35 and 45. So just before menopause. And then menopause is somehow protective because the incidence stops going up so fast, but is not going down. So we think that the retired cell state is a state at risk, and we really we hope to be able to do something about eradicating this. With this, I really want to thank all the people that did the work. This is us. This is my bar in Rimini. I was talking to Scott about it. This is where I'm going to go back in 5 years if everything fails. Thanks to the founder, of course, thanks to the patient. All this cannot be done without patient and donor. And if guys are looking at the job in London, we are heavily recruiting. So please abandon Minnesota and move to London. Thank you.
Thank you so much for a great seminar. I would like to start with a question from our virtual audience.
Fantastic.
And then those folks who'd like to ask questions, we do have microphones in the room. The first question, has your group attempted inducing dormancy in a similar manner in other subtypes, even if dormancy is not as widely reported in them, such as triple negative? This is from Maggie Chu.
So in the triple negative situation, the situation is slightly different. So I guess what we call dormant would be a persister cell stage. People have worked quite a lot about it. But I think the paradigm there is quite different because, they're working very heavily in the neoadjuvant setting. You can almost eradicating nowadays 70% of this triple negative breast cancer. So dormancy might be there, but I think it's slightly different beast. I think where this paradigm might be translate is with prostate. I think all these hormone-dependent cancer that if you think about is 2 out of 3 new cancer, these are incredibly common. They happen with age, and they tend to happen exactly when the hormones are going down. So I don't know if this has anything to do with prostate. We've done LICAP 95, of course, and what we realized there is that we could not get dormancy as breast. What we get is extremely slow proliferation. So before we get to a progression, it took us years and years and years. So we had like 15 months of treatment, but we never get to a complete cell cycle stall. And I don't know if that is something, related to the fact that we only have 3 cell line for AR positive.
So great talk. I really like the slide you had about Jack Nicholson and the different evolution of his crazy movie characters. If you think about that differential evolution, these different paths that each cell line is taking on, in my understanding, you're growing these in giant flasks where nutrients are readily available, which is very, very different than what's happening in the body, where there's a competition for the energy resources that are available. Do you think the addition? I have two. The first question that I really have is: do you think the addition of a competitive environment where some other cell type is using those resources would create a more common pathway of evolution in these pre-cell types? And secondarily, if you think about evolution, at least at the species level, the majority mechanism of what you're seeing for development of de novo evolutionary events, such as speciation, is the movement of endogenous elements. Have you carefully looked for mobile genetic elements in these?
So for the first one, of course, besides just competition, it would be fantastic to have the immune system in this, because what we tend not to think is that these are microdisseminated cells. Probably these things will play out in microdisseminated cell in very different environment, and we don't know how that will respond to immune system. Problem is that for ER alpha positive breast cancer, we don't have a gem model. It doesn't exist. So we're working quite hard to do this in rats model now. We're of course doing the cell line xenograph now in a mind model. And again, that is tricky because if you want to keep the ER alpha positivity, you need to do with this intraductal injection in the nipple. That is something not many labs can do, but we move to the ICR, working Catherine Briskin will do that. And we're doing exactly the same. So the nutrients is an intriguing thing, but if you think about, even in this flask, the moment we take away estradiol, the serum is the same, everything else is the same. So they have massive amount of pyruvate, glucose, oxygen, whatever you want. They stop proliferating completely. So I think that it's true that there is, here the competition is not so much limited by the glucose or the classical metabolic pathway, is really the receptor driven. And we have seen, like, basically with the metabolic, they shut down completely the metabolism. Dormant cell metabolism is next to zero. So I don't know if, if we say we put hypoxia on it and we put a lot of other stress, I don't know if that completely will change the landscape. I think it's more, what might change a lot is the immunoediting. And we'll see if that's true when we get to that part. For the second question that are completely, is the ERV. So yeah, there is an inflammation signature in this dormant cell. So we thought, you know what, we are de-repressing all these ERVs. And we look with nanopore, and we look at actual retrotransposition, and we couldn't find much. But we haven't looked in enough replicate, I think. What was intriguing is that in triple negative, they see, like Celine Valeo group, see exactly the opposite. So what they see is that when they do chemotherapy for a long time, in the persistor cell, they lose heterochromatin, and losing heterochromatin is reactivating on line 1. And that might be, conceivably contributing to a genetic-driven evolution. For us, we see the opposite. We see a massive increase in heterochromatin, including G9A, that is repressing generally line 1. When we look at RNA-seq, we see there's a small subset of retrotransposon that are up, and that might explain a bit of the inflammation, even if I think that is a stress signature more than anything else. Well, we couldn't find any genetic translation to that. So there might be something, but I don't know if that is gonna be able to guide. Because remember, we have one barcode. We should have been able to really see with one lineage if there was some genetic event.
There's a mic on your side. And let's just have this be our last question from our audience in the.
Thank you for a beautiful talk. So how do you know how your 3 genes are regulated, that your epigenetic modifiers, are they perhaps regulated at the transcriptional level by NF-kappa B signaling, which would make sense?
We don't know that at all. So we know that our at least EZH2 is a cell cycle regulated gene. So I think there's a bit of confusion to try to understand if this is a cell cycle arrest driven phenotype, is a truly epigenetic phenotype. I think we need to do a much more careful experiment to disentangle that, but it's not going to be easy.
And then are the ER positive retired cells in older women, would you predict that cell population would be proliferative in response to estrogen?
Yes. And I think there is a bit of argument if you look at hormone replacement therapy. If you look at the estradiol only, not the estradiol progesterone, that seems to be preventative of breast cancer.
Right. I was going to say it might explain the Tilly.
Yeah, so I wonder if a bit of that is as we keep them busy for longer, because my fear is that if you have a receptor in the cell doing nothing, and we know that if you don't have estrogen, also the estrogen receptor is a bit transcribed at higher level. At some point, it's going to find out something to do. I don't know. I have this fear of I don't trust my body. So I think if we could kind of take these cells out in a way that is not surgery, that probably should improve. Should have some sort of chemoprevention, but doing the studies, as you know, is
Well,
complicated.
presumably the breast has undergone involution because these women have finished their reproductive years, so there must be less tissue, right? But it's still highly responsive tissue, is what you're saying.
Yeah, and we see, if you look at this classical immunopathology, you see all the alpha positive. We don't see Ki67. It was quantified. We don't see it at all. But it would be curious to see what happened in women with HRT and see if they're now Ki67 is a bit back up.
Great, thank you.